Hi, my name is Jordan, and I'm going to be going over pH-induced antagonism of experience with cephalothin in combination on Klebsiella pneumoniae, with my mentor being Dr. Ellis. The rise of drug-resistant pathogens presents a major problem in clinical environments, which restricts current treatment options. Over the next 30 years, the number of deaths attributed to antimicrobial resistance is projected to nearly double. The bacteria used in the study is Klebsiella pneumoniae. It's a biofilm-forming bacterium responsible for nearly one-third of all gram-negative infections. Biofilms shield bacteria from immune responses and antibiotics, enabling persistent infections and enhancing bacterial survival. The misuse of antibiotics have driven increasing resistance in Klebsiella pneumoniae. Certain strains show high resistance to second and third strain cephalosporins, and resistance to carbapenems, which are usually a last resort antibiotic. A new approach to combat drug-resistant biofilm-forming bacteria is to combine antibiotics with a biofilm-disrupting agent. This enhances antibiotic penetration, where the biofilm-disrupting agent breaks down the protective biofilm matrix, allowing for antibiotics to reach and effectively target embedded bacteria. This reduces bacterial resistance by breaking down the biofilm and improving overall treatment efficacy. This also helps eliminate persistent infections by clearing bacterial colonies that are often resistant to clear with a single agent. The bacterial disrupting agent used in this study is XPERIENCE, Advanced Surgical Irrigation. It's a new solution designed to disrupt biofilms and inhibit bacterial growth. It's composed of citric acid, a chelating agent, sodium citrate, a pH buffer, and sodium lauryl sulfate, which reduces surface tension and lyses bacterial cells. The objective of the study was to investigate the interactions of XPERIENCE in combination with cephalothin on Klebsiella pneumoniae. We wanted to know if XPERIENCE and cephalothin work together in combination on a biofilm-formed bacteria, and can they cause an increased antibacterial effect. However, the preliminary results suggested that XPERIENCE and cephalothin at certain combinations surprisingly did not produce a synergistic effect, but rather an antagonistic one. The objective then shifted to identifying which components of XPERIENCE were driving these antagonistic interactions. The first experiment conducted in the study was to find the MIC of XPERIENCE and its components, where the MIC stands for the minimum inhibitory concentration. That means that we were trying to find the lowest concentration that inhibited bacterial growth. We did this by doing a two-fold serial dilution, and then we plated each of these dilutions into a 96 well plate. Then we added the bacterial species Klebsiella pneumoniae 43816 and incubated it for 37 degrees Celsius for 24 hours. We then obtained the optical density to quantify bacterial growth. The 6 solutions tested were standard XPERIENCE, XPERIENCE without SLS, XPERIENCE without citrates, 10% citric acid, 10% sodium citrate, and the antibiotic cephalothin. Dilution factors ranged from 1 to 1 to 124, or 1124. The results for experiment 1 are shown in figure 1, where each different color bar is the different solution tested. The black horizontal bar represents the average bacterial growth with no antibacterial solution. The MIC for XPERIENCE alone was determined to be 1 to 8 dilution. Then 10% citric acid's MIC was determined to be a 1 to 32 dilution. Then XPERIENCE without SLS was found to be a 1 to 4 dilution. The 2 solutions, XPERIENCE without citrates and 10% sodium citrate, failed to inhibit bacterial growth completely and actually increased bacterial growth compared to the control. In experiment 2, we're observing the interactions between XPERIENCE components and cephalothin at many different dilutions and concentrations. So we're using a checkerboard MIC method, where we're going to combine 11 different concentrations of cephalothin with 8 different concentrations of XPERIENCE or its component on one 96 well plate. This will give us 73 unique combinations, where if these combinations have a larger MIC than each solution alone, that will result in antagonism. And if the MIC is lower than the solution alone, that will result in a synergistic effect. We are now able to determine if this is a synergistic or antagonistic effect based off our experiment 1, where we determined the MICs alone for each solution. So for the results of experiment 2, we found that there are certain ranges in each experiment that results in the antagonism. So for standard experience, these antagonistic ranges were the 1 to 16, 1 to 32, and 1 to 64. And these can be seen with these optical density peaks around 3.5 to 4 or 0.4. And these numbers above each bar represent the pH of the solution at that dilution. So there's a clear pH range that is resulting in this antagonistic effect, where that is around 4.252 to 5.13. And I should add that each color of the bar represents a concentration of the antibiotic cephalothin, where 16 represents the MIC, so we wanted to add in a 1 to 2 dilution factor above and below that MIC. The second solution from experiment 2 that had a clear antagonistic range was 10% citric acid, which had a antagonistic dilution at 1 to 32 and 1 to 64. We didn't include 1 to 128 as an antagonistic dilution because there's growth at a concentration below the cephalothin's MIC, so that's not included as an antagonistic range. If there was growth at 16 micrograms per milliliter, then there would have been, but not in this case. But there's also kind of a similar pH range for 10% citric acid that inhibits the cephalothin from working, and that is 4.2 to 4.72. The same thing could be said about experience with SLS, where at the 1 to 8 and 1 to 16 dilutions, there is a clear antagonistic range, where at the pH is 4.54 to 4.84, they actually inhibit the cephalothin activity and just the total antimicrobial activity of both solutions alone. That 1 to 32 dilution, though, similar to 10% citric acid, we didn't count that as an antagonistic range since the growth was only at the a level below the cephalothin's MIC. So now, after comparing all three of these graphs side by side, you can see that each of these antagonistic ranges are actually due to a pH factor and not a experience component factor. So we didn't actually include 10% sodium citrate or experience without citrates, since those solutions in combination with cephalothin did not inhibit bacterial growth at all, so we couldn't see any clear pH or bacterial growth peaks since every solution grew. But as the solution antibacterial agent is diluted, the pH of the solution increases, and the pH ranges of 4.2 to 5.13, experience inhibits the antibiotic activity of cephalothin, resulting in an increase of bacterial growth. The key findings for the study was that there was an antagonistic interaction observed between the experience components and cephalothin, and that actually resulted in a reduced antibiotic efficacy at these certain pH ranges, and these ranges were 4.2 to 5.1. This highlights the role of pH in antibiotic efficacy, where pH should not be forgotten about when combining biofilm inhibiting solutions and antibiotic. These combination therapies need to account for pH optimization and specifically against Klebsiella pneumoniae. Further studies should use other antibiotics and also other biofilm and disrupting agents. Also, these solutions should also look into pH-adjusted formulations to maximize their efficacy as they get diluted down. In conclusion, this study found that experience effectively disrupts biofilms and eliminates Klebsiella pneumoniae, specifically 43816, up to a 1 to 8 dilution. However, when combined with cephalothin, instead of a synergistic effect, It actually antagonizes the antibiotic at mid pH ranges 4.2 to 5.1, leading to increased bacterial growth. This antagonism was not caused by any single component of XPERIENCE, but rather an overall pH of the solution. These are my references, and I would like to thank Dr. Ellis for her guidance throughout this project.